And with that, I will introduce the first talk of the conference. Jonathan Weissman is Clay Professor of Biology at MIT and at the Whitehead. He's a member of HHMI and a member of the National Academy. As many of you might know, Jonathan's work has really centered around trying to understand protein folding within the cell. And in order to address these questions, his group has been motivated to develop many technologies to look at this problem in new light and new dimensions. And those technologies have been very broadly applicable for many cell biological questions. And so probably many of you in the room are familiar with a few of the tools that his group has created. And we're really looking forward tonight to hear about the latest. Thanks, Jonathan.
All right, you can hear me? Well, thanks so much for the kind introduction, and especially for the invitation to be here to speak to all of you. I'm really looking forward to the rest of the conference and want to tell you today about some of our work on epigenetic editing. And because I have a little more time, I'll start with a bit of history that at least to me was fun and interesting, which is my lab. Okay, it's not moving forward. I am pressing the green button. Can you manually move it forward for me, or someone? Let's see. All right, so my lab's interest in CRISPR, I would say, like many people, CRISPR was a curiosity for a while. And then that really changed in the summer of 2012 when I had three very propitious people walk into my office. So the first was Jennifer Doudna, who I'd been talking with about her CRISPR work for a while. And this was just a couple months after she had published that Cas9 plus a single guide RNA was a programmable nuclease. She came in and sort of said, we have this great programmable nuclease. Think about what are all the things that we can do with this. So that was a fun challenge. And then the next was Stanley Chi, who was then actually finishing as a graduate student at Berkeley and coming over to UCSF as a fellow. And he had taken Jennifer's dead Cas9 and shown that it could block transcription in bacteria. And then the third was Luke Gilbert, who had just joined my lab as a postdoctoral fellow and had a ton of experience in sort of the lentiviral vectors and sort of mammalian cell biology and delivery. And so between this, the sort of four of us and several others thought, well, the perfect thing to do with this beautiful programmable nuclease would be to break it. So, all right, I'm going to have to tell you, please advance. So, to break the scissors. And the reason why, of course, is, next slide, is that if we do that, so advance. If we do that, we would now, instead of having a programmable nuclease, we in theory had a programmable DNA binding protein. And then you could make fusions to that, bring in effector domains. So, for example, a transcriptional repressor or a transcriptional activator. And in this way, advance. In this way, we could tune the expression level of any gene or any sets of genes. And this we thought would be a very useful tool. And since this time, there's been kind of a remarkable array of different effectors, way beyond what we could have anticipated, including things like deaminases that David Lewis so famously used, or reverse transcriptases. So this has proven to be quite general. So next slide. Okay, I need and just advance one more. Given. Sorry. All right. So that this allowed us to do, in pooled screens, to put in libraries of different guides, each targeting a different gene, and in this way, for each cell, have a different genetic perturbation. And then one can do a screen where you look, for example, which genes, when you knock down, confer sensitivity or resistance to a drug, or which, if you use a fluorescent reporter, which turn on, for example, a stress response. And this has been broadly useful to us and to the community. Next slide. So the challenge is that when you're doing this, by definition, you're only getting a single one-dimensional readout. So you can get the growth, the effect on growth, or the effect on the induction of a stress response. But two things. So then you have to predefine what you want to look at. But also, of course, and please advance, that cells exist in a much more complicated high-dimensional manifold. And essentially what you're doing is doing a very complicated, poorly understood dimensionality reduction to this one dimension. And you can have two cells that die, for example, for very different reasons. And yet, if their level of their growth defect is the same, they would score the same by this type of approach. Please advance. And then one more. One more. Stop. All right. So these were over-animated if I was not going to control, but I'll do my best. So this motivated us, and working with Aviv Regev and a couple other groups in parallel, to develop the so-called perturb-seq approach, in which you would, again, take a library where each cell had a different perturbation, but now you would do a single-cell RNA-seq experiment. And in that single-cell RNA-seq experiment, you would read out, as you do in single-cell RNA-seq, the state of the cell. But in addition to reading out the state of the cell, you'd read out the nature of the perturbation. And this essentially allowed us, for each perturbation, to see where in this high-dimensional space, in what direction that perturbation is putting the cell. And we and others have used this really quite broadly to explore gene function. And in particular, in two papers, I'm particularly proud of. The one from Tom Norman. We used this as a way of looking at genetic interactions and showed that it was far more interpretable to look at genetic interactions when you had a high-dimensional phenotype to read out rather than just growth, live death, for example. And then a second paper we published a year and a half ago or so, published the first genome scale perturb-seq, and that gave us our first sort of large-scale genotype-phenotype maps, and showed that you could then use this to systematically probe after the fact, sort of in silico create phenotypes and probe the data set to understand what the genetic basis of these. And we're particularly excited about these large-scale experiments. And I think just in the last year, the scale that we can do this has increased by 1 and really close to 2 orders of magnitude. And we think that this is going to be, and also where the type of cells we can study, in particular being able to start to think about doing these types of experiments from in an in vivo setting. And we think that these are going to be particularly valuable data sets as we and others try to train transformer models to get to get a foundation model that really learns the core underlying functions of a cell. So all of this was terrific from a discovery point of view, but if you're starting to think about a therapeutic control of gene expression, the CRISPR-RNA and related approaches weren't very useful. The reason why was they're transient in nature. So you would be able to knock down gene, but only as long as you could overexpress your complicated Cas9 protein. And this motivated us to develop the CRISPR-off approach, which based on methylation of DNA rather than sort of making modifications of the histone. Methylating the DNA allow you to permanently silence genes. And the reason this was possible was piggybacking on a lot of highly evolved gene regulatory, gene silencing mechanisms, both for silencing genes during development and for, in particular, the system that we we exploited for silencing parasitic elements like endogenous retroviruses, as well as ways of identifying these and making sure that they don't express. And one of the reasons these are so robust is that in addition to the de novo methyltransferases that methylates a C at a CG sequence, there's a maintenance methyltransferase, DNMT1. And what DNMT1 does is if the DNA is replicated, so you're hemimethylated, it takes this hemimethylated state and makes sure that the second strand gets remethylated. So with this and building off beautiful work from Angelo Lombardo, we built all-in-one DCAS9 with 3 domains, a CRAB domain that does, among other things, H3K9 methylation, so the CRISPR-I repression, 3L, and then the 3L and 3A domains, which are responsible for the DNA methylation. And after, and I say this work was led by James Nunes when he was in the lab, who's now running his own lab at Berkeley. And what he was able to show after a lot of tweaking is that now you would have a transient expression of the CRISPR-off, but instead of like CRISPR-A, where the gene would reactivate as soon as the epi editor was gone, now you could have quite stable silencing. And in one experiment, James carried this out for 15 months, passaging the cells continuously, and even after that, the vast majority of cells were completely silent. So it's exactly unlike this analog rheostat that I showed to begin with. This is a switch, and when you switch it off, the cell's able to maintain this. And now, more recently, we've seen work from people like Angelo Lombardo that's shown that this can be maintained for really extensive times in an animal. So, and it's exquisitely specific, so we're really just hitting the one gene that we're targeting. So, in full disclosure, I, with Luke Gilbert and Keith Young and David Luce, started a company, Chroma, whose goal was to use epigenetic editing to silence disease-causing or disease-promoting genes. So I thought Chroma would go and do the therapeutic, which is something didn't know anything about, and we would use the epi silencers as a way of studying the mechanism by which epigenetic marks or DNA methylation is initiated, propagated, and even reversed. And then, sort of almost 10 years to the date after Jennifer walked into my office at UCSF, Sonia Valib and Eric Minical walked into my office in 2022 and said, we want to write a grant that will fund, and that's a 5-year grant whose goal is to bring us to an IND-enabling study to silence the human prion protein. And just, the important backstory of this, they were highly motivated to do this because Sonia actually had inherited a fatal familial insomnia mutation, a genetic form of prion disease, and was a Harvard-trained lawyer, and she and her husband Eric, when they found this out, quit what they did, went to graduate school, and now have a lab at the Broad, whose goal is to find therapeutic strategies to prevent and reverse prion diseases. So, to remind you, the prion protein is a natural endogenous protein. It doesn't seem to be required. It can be knocked out in multiple different animals with only at most modest effects. But it can, at a very low rate, promoted by some inherited mutations like the FF5 mutation I talked about, it can convert to self-propagating aggregate. And these aggregates then lead to a rapid decline and death. And it's very well recapitulated in the mouse. And the key thing is from work starting with Giovanna Malucci, and has been done in many different contexts, that we know that turning off the prion protein, specifically in neurons, even in active disease process, even in an animal that's starting to become sick from the propagating prion, will halt and even reverse the disease. So from a biological perspective, this is sort of the perfect target. We know we can get rid of it, and we know that getting rid of it will really have a therapeutic benefit. So then the challenge really was, could we silence the prion gene? And so we started working in cell lines, it has a nice CpG island. You can methylate it nicely, and just like the data I showed you previously, you methylate it. CRISPR-I is reversible. CRISPR-off, it propagates indefinitely, and it does this because it fills in the CpG island with methylated CpG, which then are propagate indefinitely. So now the problem is really just one of delivery. And here's where Cas9 is really not well suited for this. Cas9 is a very large protein, and basically the best delivery approach is using an AAV virus, which has a payload, a limit of about 4.5, maybe 5 kb. And so Cas9 is, if you want to do something like a base editor or a prime editor where you really need to nick or you need the specific bubble, Cas9 is very well suited. But for this problem, it did not seem to be particularly well suited. And in fact, an older technology, the Zinc finger technology, was much smaller. It didn't open up the strands, so it really would put the effectors right on the DNA. And in fact, Zinc fingers, a very common thing that they're by evolution fused to is the crab domains, and they're used broadly for this type of silencing of endogenous retroviruses. So, and just graphically, just to give you a sense of this is what CRISPR-off looks like compared to the AAV genome, and this is what a ZFP-off version of this would look like. So we would have plenty of room to play around with. So this seemed like a straightforward shot. But then we got a rude awakening, which is when we made ZFPs, the ZFP-off versions, and transiently transfected, they're actually rather toxic to cells. And this was specifically due to the DNA methylation domain, the DMT3A methyltransferase domain. So why is DNMT3A toxic? I have to jump into a little bit about the details, but I think it's sort of an interesting story of regulation. The DNMT3A is actually a multi-domain protein, and it exists in an auto-inhibited state. And that makes sense because I told you once you methylate DNA, it can propagate. So it would be sort of like having a mutagen if it were there constitutively active. And the way we and others have gotten around this is we fuse just the methyltransferase domain, which removes the auto-inhibition. So you now have a constitutively active methyltransferase. And whereas normally the full-length 3A would be in an off inhibited form, and only when it sees the things like the histone tail that can release this auto-inhibition is able to turn on. And we also knew from a very nice study, a number of studies, but one that really motivated us was from 2022, that just adding in vitro this histone tail peptide to DNMT3A was enough to activate the full-length DNMT3A. So this led Edwin Newman, who was just starting his second year in graduate school and now is a third-year student, to propose that we could just take a Cas9 3L fusion, and that would recruit the endogenous DNMT3A. But of course, it would recruit it in an inactive form. But if he now added and got the geometry right, the histone tail, it could recruit and activate the methyltransferase and allow silencing of genes without actually having to overexpress any enzymatic activity. So Edwin made a variety of different fusions along these lines. And so CRISPRoff is sort of the thing we have to benchmark against. The CRISPR-I is the transient in nature. The 3L alone had some activity, as others had shown, but was highly compromised compared to CRISPR-off. But if you added the histone tail, it now, even in these early experiments, and I'll show you later, much more so, is able to compete with the CRISPR-off. And we also knew that we had crystal structures of the histone tail bound to DNMT3A, and the prediction was that this K4 would be absolutely critical for this interaction. So Edwin repeated this experiment, but with the mutant histone tail that had replaced the K4 with an alanine, and that completely abrogated this effect. So he was pretty confident that not only could he activate the endogenous, but he really understood how it was activating. So this worked like a charm, and so thus its name, coupled histone tail autoinhibition release of methyltransferase, or CHARM. He made a variety of many iterations of this. A key one was screening through different 3L domains from different species, and the wood mouse by far actually won out over the rest. And with this, he was able to get variants of charm that were consistently as good or better, in most cases better than our original CRISPR-off. So this also, unlike the 3A, 3L, the ZFP-off version, we didn't see this toxicity. It was highly effective at silencing when we used a zinc finger targeting the mammalian prion promoter. It was highly effective at silencing this, and it's silenced through methylation. So this is sort of the typical picture you see, where most CPGs are methylated, but then around a promoter of an active gene, you have an unmethylated CPG island, and the ZFP-off, or charm, is able to basically just fill this in and silence the gene. So with this, he was now in a position to start thinking about doing the actual mouse experiment. And we teamed up with, so I should say, Eric and Sonia's group really led all the mouse experiments because they had all the infrastructure for doing this. And Ben Deverman, our partners on all of this, led the AAV. So we made the AAV. We designed our first AAV for Ben and sent the design over. And he said, "This is great, but could you make the insert a little bigger? Because it's so small, you might end up picking up random DNA." And any of you who've been in the game of trying to make things fit into AAV would appreciate that was an unusual request from someone who's making AAV. But also the small size made the efficiency of making the AAV much better. And basically from the first experiment, I should say this is a couple iterations in, but from the first experiment this really worked beautifully. So we were able to, across the entire brain, even though we know we can't hit microglia and they express prion protein, but grossly across the entire brain, we were able to get 80 to 90% reduction in expression of the prion protein. We also knew from ASO work that 50% and even as little as 25% reduction was sufficient to start to interfere with the disease process. And this also methylated the prion gene in vivo, so these are from the brain extracts doing nanopore sequencing of the prion locus. And then, but really want to prove that this was happening in neurons, and you can see just grossly across the brain, we're ablating prion expression, and when you do this quantitation, even within neurons itself, and we knew it was sufficient to knock out PRNP in neurons, the vast majority of neurons are essentially completely off. And this was also important because I told you DNMT1 is this maintenance methyltransferase that's coupled with DNA replication, but a neuron's not dividing, so you don't have that mechanism. So it was not a foregone conclusion at all that we would be able to see this type of methylation in vivo, and we're really delighted to have that. So then the final thing we thought, well, since we're doing this hit and run methylation approach, we don't actually want a continued expression of the ZF charm because we knew it was well tolerated on the period of weeks that we were looking at this, but how do you de-risk something if it's going to then, the AAV is going to continue expressing this for the rest of the life of the animal? So this led in work that's been led by a terrific postdoctoral fellow in the lab, Tessa Bertozzi, to develop a self-silencing approach in which you would have the ZFP charm that was then silencing your target, but then also could silence the gene itself. And the way we made an allelic series is we either had a scrambled site, a single point mutation that compromised the binding, a perfect match, or a double perfect match. And what she found with this is, thankfully, on short time scales, they all very nicely silence the prion target gene. But then when we looked at the charm, in the one where it scrambled, it was on its short time scales, but even at very early time scales, the one with the double perfect match binding site in the promoter of the ZF charm was quickly turned off. And then when we looked after 2 months, we saw that sort of the single mismatch and the perfect match were really nicely silenced, but the double perfect match was self-silencing too fast. So this is sort of the Goldilocks experiment, and I guess in that paradigm, the mama bear is the single perfect match. And so now we went into a mouse with these self-silencing vectors, essentially like the first experiment, but now all we did was put either double, perfect match, point mutation, or scrambled upstream of the promoter that was expressing the charm. What we saw is, again, this really nice allelic series where the scrambled point mutation did the same. The perfect match maybe was a little bit compromised, but still did very nicely. But the double perfect match didn't silence nearly as effectively, and it didn't because it was self-silencing too quickly. So this is actually looking at the methylation of the AAV vector itself and seeing that as we ramped up the number and the efficacy of the binding sites, we were getting better methylation of the AAV. So the notion is that we have, there's still some refinement to do for sure, but that we basically have a system where you transiently express this programmed DNA methyltransferase. It shuts down, methylates permanently, shuts down and silences endogenous gene, and then within 6 weeks it silences itself, and it's essentially just a neutral piece of DNA, not expressing any protein. So just to summarize, we think that the charms are going to be very, in many ways, an ideal DNA methylation system or silencing system. They're smaller than CRISPR-off, so essentially for any of the delivery modalities, whether it's LNP or AAV or viral-like particles, it's going to be easier to deliver them. They don't have two parts, the guide RNA and the protein, which means you don't have to get the stoichiometry right and makes the delivery makes the formulation much easier. They're all made from human parts, so they shouldn't be immunogenic, especially if transiently expressed. That's in stark contrast to Cas9, which many of us have naturally antibodies against. And then, so we're very excited now we have a humanized mouse and a human version of this, and shown that we were able to silence the human prion protein promoter. Now, so we're very excited to move forward these projects forward with Sonia, Eric, and Ben's lab. But also, I think just more generally, when you think about the future, as delivery is really the limiting thing for almost all of the genetic medicines. And this, we think, is going to be sort of the ideal molecule for many delivery modalities. And although there are therapies like genetic mutations where you really want to be able to change the DNA, by far the most common mode of therapeutics is as inhibitors of existing proteins. So for that broad class of therapeutic targets, we think that the ZFP charms or other variants of charms may be ideally suited. So with that, I'll thank you guys for listening, and especially thank the people who did the work. As I said, this has been just a wonderful collaboration with Ben Deverman, Sonia, and Eric, and led by Edwin on the charm, and Tessa on some self-silencing work, and a really spectacular technician, Elaine Wu. And Fiona Sarek leading the mouse work with Sonia and Eric, and then many other people along the way. So thank you, and I'll take questions.
Does it go into the germline? Have you tested this on the mice?
The AAV is unlikely to. LNP certainly can. We think it's very unlikely that it would propagate, unlike a change in the DNA. So people, for example, are very concerned with PCSK9, or the FDA, I should say, is because there's LNPs to deliver, but they also do go to the germline, and you don't want that modified PCSK9 passing on to the next generation. Whereas, of course, as you know, there's waves of demethylation after fertilization in blastula. Yeah. So but we're doing this. We'd really like to actually just briefly epigenetically silence every cell in the mouse, see how long it perdures, and then also see if it goes to the next generation. So if there's really transgenerational, it would be quite spectacular, and there's a lot of evidence to say that it's unlikely to, but if it did, it would be a great finding, but I think we'll be able to, most importantly, we'll be able to answer fairly definitively, and likely that it's not going to.
Yeah, that was beautiful, super exciting work. I was wondering if you already started to evaluate how portable it's going to be to other promoters? Because I understood this one is a CG-rich promoter. Mm-hmm. Is there a dependency to CG density on the system? Did you started to play around with this?
Yeah, so our feeling, so it has, the human technically has something called a CPG, would be called as a CPG island. As it turns out, the mouse is sort of below the threshold, but does have some CPG. So most promoters have more CPGs than the mouse, and there was no problem silencing at all. So James Nunes, with the CRISPR-off system where it was programmable, did genome-wide screens against all the essential genes, and he showed that he was able to get silencing at the vast majority of the essential genes, even many that weren't called as having a traditional CPG island. Now, if you have few enough CPGs, they either methylate or sometimes they reverse, and we're really interested, and sometimes there's like really ones that you'd like to do, like ANGPTL3. So PCSK9 is really easy to methylate. ANGPTL3 is one that is actually seems to demethylate. So we'd really like to understand, can we switch this over? But in terms of going from CRISPR to ZFPs, which are, of course, harder to design, I think if you can flip it over with the CRISPR system, it's very unlikely that you won't be able to do it with the right zinc finger. So that's why we sort of started by the very beginning. We showed we could methylate it, the promoter, and it would propagate, and then you can move forward. Yeah.
Self-silencing is really cute trick, and you said that 2 copies was too much. That obviously shut it down too fast. What I'm wondering is, could it be anything about the position of the copies rather than just the fact that there are 2 of them?
For sure. I mean, we just put them around some CPGs. We didn't really spend any time optimizing that at all. And we've been working with Mo Khalil some, and because we can actually fit 2 or 3 zinc fingers, we could have one that's like tamoxifen controlled and dedicated for self-silencing, the other one that's going endogenous. That way you could wait a defined period of time, give tamoxifen that would allow it to go into the nucleus and self-silence. So we think that each one will require some engineering, but we think that there's a lot of levers to pull on that.
This was very impressive, and it's clear there is enough efficiency for the therapeutic application. I'm just curious if you have an idea of, or a lead on the escape cells that are not silencing. What are the possible mechanisms?
Ben's perspective is it's amazing that we're hitting this fraction of the neurons that the AAV, he's never seen the AAV actually hit this well, and I think it's because only one first of all, the genomes are small, so the AAVs you get a higher, tighter, less empty ones. But our feeling is pretty much every cell that's getting the AAV is probably silencing. So in a sense, the silencer is perfect and is really a delivery problem. Now I should say the AAV delivery to the mouse brain crossing the blood-brain barrier is a little bit specific to the mice, but there's big efforts to get AAVs that can cross the blood-brain barrier in humans, including one that Ben just posted a few months ago that uses the transferrin receptors to pass through. So I think that's at this point the AAVs are the weak link, but there's a lot of work going on there.
Okay, let's thank Jonathan again for that wonderful talk.